In this lecture, we will focus on proteins and their monomers, which are amino acids. We will start by talking about many different functions of proteins. They can do all sorts of things. Then we will specify that amino acids are the monomers, and we will talk about the general structure of amino acids. We will also see that there are about 20 different common amino acids, and what makes each one different from the others is the atoms in their R group. What the R group is the variable part. And we will talk about how we can determine general properties of amino acids from what is in the R group. Finally, we will talk about how amino acids are strung together into polypeptides and how different levels of structure in the polypeptide determines its shape, and how the shape then determines its function. So let's start by talking about the function of proteins. Proteins can do many different things, and those functions are categorized differently in different sources. But here's one list that attempts to break down the functions. One such function is having a regulatory function. And an example of this would be a protein hormone, such as insulin. And a hormone is going to be a molecule that sends a signal a long distance through the body. We'll talk more about that later in the semester. And so insulin is going to send a signal telling various cells in the body to reduce sugar supply in the blood. For example, by pulling sugar out of the bloodstream and into various cells. Another function of proteins is structural. And so an example of this is collagen, which is a protein that occurs outside of our cells, so attached to the outside of the cell membrane, and it helps connect cells together, for example, to give your skin extra strength. A third function of proteins is protective, and this can take different forms, but one such form are antibodies, which are part of the immune system in the body, and they are able to attach to various chemicals that may occur in the body to help the body identify that they are there, so they can then be dealt with. A fourth function is transport. And we will see examples of transporters within a cell or in the cell membrane later in the semester. For now, I'm going to show you a hemoglobin molecule, which is here. And hemoglobin, of course, carries oxygen in our blood. So I can draw an oxygen on here. And so it transports it from our lungs to cells within the body. Another function is enzymes, and enzymes are typically proteins that accelerate chemical reactions. We will see many examples of this throughout the semester, but one such example is the lactase enzyme. And lactase helps break down lactose. So lactose is a disaccharide that we talked about with regard to carbohydrates. And as part of our digestive process, if we are to use these sugars for cellular functions, we need to break this bond right here and do hydrolysis to split this into the two monosaccharides. And one of these monosaccharides turns out to be glucose, which our bodies can use easily. The other one is galactose, which our bodies would convert and then use as an energy source. Another function of proteins is for defense. And one such function is making toxins, such as the toxins made by a black widow spider. That could, of course, it could inject into the body of an insect or a human if it was either trying to eat or trying to defend itself. And a final function of proteins that we'll talk about are proteins for movement. And one such example of this are the actin and myosin proteins. So we can see actin in the green up here. Oops. And myosin is this larger structure down below. But actin and myosin will move against each other, so in this direction and this direction, causing muscle contraction. And of course, muscle contraction then allows animals to move. So those were just examples. And there are other ways of dividing up this list, but this gives at least a general sense of the many things that proteins may do. Let's focus on enzymes for a minute and just say a little bit more about them. We've already said that they speed up chemical reactions, and we have a term for that. Any substance that speeds up reactions can be called a catalyst. This just literally means it will go faster. And enzymes are typically proteins that do this, and we can say that enzymes are highly specific. So different proteins, depending on their shape, will catalyze different kinds of reactions. I showed you the lactase enzyme just a minute ago. Here's another example of a different enzyme that converts a different disaccharide, shown here, into two monosaccharides, shown here. We will talk more about enzymes when we talk about metabolism in just a few chapters. Here is a summary list of those different functions. We can see that there are transport, structural, regulatory, defense. This says contractile. We can call this movement. We can also add storage as a function of a protein. This is typically storing amino acids so they can be used later for other purposes. So that can be added to our list. And then digestive enzymes is really just a subcategory of the more general category, enzymes. Enzymes do lots of other things too. Let's now talk about amino acids, which are the monomers of proteins. And so you know the monomer is, by definition, the repeating subunit that makes up the polymer. There's a general structure to amino acids that all kinds of amino acids follow. And then there's a variable part that give different amino acids their different properties. So let's start by looking at the parts that are always the same. First, there is the central carbon atom. And that is right here in the middle. And it has 4 different things that are always attached. First, there's always an amino group attached, and you know that is NH2, pictured here. There is always a carboxyl group attached, and you know that's a carbon double bonded with an oxygen and also attached to a hydroxyl group, which I just circled there. There's always a single carbon, easy to forget, but it's right here. And then there's the variable part. So the R here just means really anything that you want to attach, or that the cell wants to attach. And there are 20 different things that cells typically attach to the basic structure. We'll see that it's the side chain, or the R group, that gives amino acids their unique properties. So as we just said, there are 20 different common amino acids. There are a few more that are much less common. Each amino acid, each of these 20, has a different R group, and the R group determines the chemical nature of that amino acid. Even though all 20 are a little bit different, we can generally categorize them into one of 4 categories. One category, and I'm going to start second down, is charged R groups. So they have either a positive or negative charge associated. A second category is polar R groups. So there's not an absolute charge, but some portions of the R group have a partial positive charge, and some portions have a partial negative charge. For both of these, polar and charged, we know that these characteristics are going to be associated with hydrophilic or water-attracting properties. And you should understand from chapter 2 why that is. Third, we can have nonpolar R groups. And those nonpolar R groups can have two different general structures. We can have nonpolar R groups that are relatively linear in structure, so there are no rings, or we can have nonpolar R groups with rings, and we refer to those structures with rings as being aromatic. And that's because ring structures, oftentimes in other contexts, have an aroma or a smell associated with them. And so we'll see some examples of what those rings look like momentarily. Here's a chart of all 20 amino acids. You do not need to memorize this chart, but you do need to be able to find an R group if I give you a general diagram. And you need to be able to figure out the general characteristics of that amino acid based on its R group. So if we look at this top left section, we can see that all of these R groups are completely or primarily carbon and hydrogen. If that's the case, we know that carbon-carbon bonds and carbon-hydrogen bonds are both connecting atoms with similar electronegativities. Therefore, these should all be nonpolar. And we can also see here that we don't have any structures that have a ring-like structure. Let's now go all the way down to the bottom right corner of this diagram and look at some more R groups. Down here we see structures that look like rings. And there's not a lot of details shown, but the reason for that is that whenever there's an intersection that's unlabeled, we know it involves a carbon. We can write those in. And whenever we see that a carbon is lacking some atoms that should be bound to it, we can assume that there are hydrogens bound. So I can add some hydrogens in here too, like so. And so now we know that this ring is really made out of carbon-carbon and carbon-hydrogen bonds. If that's the case, then we know that these rings should be nonpolar. Now let's look at a couple of other categories. Down here in the bottom left corner, we can see R groups that don't have any charge associated, but we can see combinations of atoms that we know are polar. For example, we know that oxygen and hydrogen differ in electronegativity, and so this is going to give us a polar portion to this R group. And we can identify similar R groups or similar polar regions in these sections I'm circling. And here, and here. So all of these R groups are going to tend to be polar, even though they are uncharged. There's no absolute charge. And we're just going to simplify, ignore that plus there, to make our life a little bit easier. Additionally, we can look at two final categories. In both of these categories, what we can see is that there's an actual charge. So there's these pluses here, and then down in the next category, there are these minuses. So these aren't partial charges like in a polar molecule. These are complete charges. And so if these are charged, then of course, these are going to be hydrophilic. Additionally, in some situations, the negative charge in one amino acid might be attracted to the positive charge in another amino acid. So these amino acids are going to be connected together in long strings. And if somewhere on the string we have a positive charge, let's say right up here, and elsewhere on the string we have a negative charge, then those charges can attract to each other, and that can influence the bending of the molecule, and it can create a bond, in this case an ionic bond, between two different R groups that will help keep the shape of the molecule in place. So that's part of why these positive and negative charges are important. Amino acids need to be represented. They each have a name, and those names are shown on this diagram, glycine, alanine, and valine, for examples. In practice, those can get long and cumbersome to write, so we use abbreviations. We often will use 3-letter abbreviations or sometimes 1-letter abbreviations. When possible, that 1 letter is just the first letter of the amino acid, but sometimes that doesn't work because there are too many that start with the same letter, and so sometimes it starts to deviate. You do not need to memorize abbreviations or 1-letter abbreviations. Just know that they exist. For humans, amino acids can be broken down into 2 categories. There are those that we must eat in our diet because our bodies have no way of making them. And those are called essential amino acids because they are essential for survival or health. Then there are also the non-essential amino acids. We still need them, but we are able to modify other amino acids into them. So if they're not a big part of our diet, we still have a way of producing them internally. Now, the different R groups on amino acids are important because they determine the properties of those amino acids. And we will end up seeing that the sequence of amino acids in a chain, the length of the chain, or in other words, the number of amino acids, those 2 things come together to influence the protein's shape, of course its size, and the size and the shape of the protein is going to control its function. So what it does is determined by its shape, and its shape is really determined by the organization of the amino acids within the polypeptide chain. Let's talk about how amino acid monomers become peptides, or chains of amino acids together. They are joined together by the process of dehydration synthesis, which we have seen now. We've already seen this for carbohydrates, and we've seen it in forming triglycerides. We're now seeing it a 3rd time. And you can see the atoms in red are going to be the ones that form a water molecule when dehydration synthesis happens. So a hydroxyl group here combines with a hydrogen on the next amino acid that forms water and is released. This carbon now becomes bonded to the nitrogen, and the resulting structure is shown in blue. This is called a peptide bond. So if we have 2 amino acids joined together, we can call that, like for example here, a dipeptide, because there are 2, and di means 2. If we have more than 2, then it would become a polypeptide. Poly means many. So a polypeptide is a chain of amino acids joined together by peptide bonds or peptide linkages. I asked what's the term for 2 linked amino acids, and we just talked about that on the last slide, dipeptide. We now can talk about what is a protein. A protein is similar but not exactly the same as a polypeptide. So a protein is either one polypeptide in some cases, or in other cases it's multiple chains of polypeptides that come together. A protein has a unique structure and a unique function, again based on the length of the polypeptide and the order of amino acids within it. And just as a heads up for when we talk about this at the end of the semester, the process of making a protein is called translation, and it's going to use RNA or a nucleic acid as the information it uses to decide which amino acids to put in which order, and hence which kind of protein is being made. So translation is that process, and we'll talk about those details much later in the semester. So I keep saying this, but here it is in writing. The shape of a polypeptide or protein determines its function. Because shape is so important, we are going to talk about 4 different levels of organization that influence a protein's shape. And these are just called primary structure, secondary structure, tertiary structure, and quaternary structure. That just literally means first, second, third, and fourth. And let's take those up in order. The primary structure of a protein is simply a way of referring to the order of amino acids in a polypeptide. So it has nothing to do with the 2 or 3-dimensional shape of the polypeptide. It's really just a list of the names in order from the start to the finish. So here the primary structure is, well, you can see the 3-letter abbreviations, and we could just go down this row. We don't have to worry about things like this disulfide bridge. In fact, we're just going to get rid of those disulfide bridges because those affect higher levels of structure. But we can see that this protein has 2 chains to it, 2 polypeptides. The first one has this particular order. The second one has this particular order, and they just happen to write it going around a corner like this. If we were to change the identity of some of these amino acids, like making that change or changing this, for example, then because these new amino acids have different properties, we'll see that would eventually change the shape of the protein and hence change its ability to function, or maybe its function altogether. So the order of the amino acids is really very important. Let's move on and talk about the secondary structure of a polypeptide. This term refers specifically to folding within small areas of a polypeptide. So if you imagine a rope or a garden hose, it might be very long, but within particular sections, it might be coiled up, for example. And so what we're doing here is just giving names to particular sections that happen to have similar shapes. Here, here, and here, there's a similar shape we might give a name to. And as it turns out, there are 2 common repeating shapes in proteins. These are called alpha helices, or singular alpha helix, and beta pleated sheets. And an alpha helix, as you might expect, looks like a helix or a corkscrew. So there's one pictured right here. Creates a spiral. And there are certain organizations of amino acids that are going to create bonds that allow this helical shape to occur. So if we were to look through the primary structure of an amino acid, that sequence of amino acids, we could identify structures just from what we already know about the kinds of orders that tend to form alpha helices, and we could figure out where along that structure alpha helices are likely to form. This is one of the first things we figured out about protein structure when we were first learning to understand it. There are other sequences that we know are likely to create beta-pleated sheets. So what an alpha helix looks like is pretty easy to draw. What a beta-pleated sheet looks like is a little bit more complicated. It looks kind of like if you think of an old-fashioned fan that you might, wave on a hot day. A fan has that shape, but of course along the top it looks more like this. There are pleats along the fan, and it helps give it some structure so the paper doesn't flop around when you're waving it. So there's these creases like so. Or if you think about a traditional skirt, a skirt that has, again, folds in it like this. So let's see if I can draw a pleated skirt. Something like this with creases and folds. Then this also has pleats. And so a pleat just refers to this up and down folding pattern. And you can see a pleated sheet drawn here in our polypeptide. This isn't just one chain of polypeptides. In this example, it's two chains on top of each other, but the chains fold back and they both are folding in exactly the same pattern. They are also creating hydrogen bonds. You can see hydrogen bonds right here. These aren't covalent bonds. They're just attractions based on partial positive and partial negative charges between nearby atoms. But that's going to hold this top layer in a similar pattern to the bottom layer, and they together are going to create these folds. And then in a further out view, here's what a beta-pleated sheet might look like in a protein, sort of in this area. So alpha helices, beta-pleated sheets are our secondary structure. Let's move on to tertiary structure. We already said that in our garden hose, or our rope, we might have some places where we have repeating patterns, like so. But then, between those places, the hose could be folded in different ways. It could continue out this way, or in another example, it might fold back over itself like this, and you could imagine it then continuing in one direction, or maybe it folds back and gets further entangled in a different direction. So these additional layers of folding that determine the final folded shape of each individual polypeptide is referred to as the tertiary structure. So this is the unique three-dimensional shape of a single polypeptide. Single is important here. And so for a protein that is composed of only one polypeptide chain, the tertiary structure is the highest level of structure. It is determined by the properties of the R groups. And so, for example, if we had a R group with a positive charge right here and a negative charge on this other strand right here, that could pull the two strands together and it could anchor this connection between them. If we had two positively charged amino acids in contrast, that could push those two portions of the polypeptide apart. So here's maybe a positive and here's a positive. So then it wouldn't fold together as it did in the previous example. We could also have hydrogen bonds hold portions of the tertiary structure together, and we can have covalent bonds hold portions together. Let's go on to the next slide to look at some of these. So here's a unique three-dimensional structure, and we can see an example of a hydrogen bond, so a non-covalent bond based on polarity, holding this portion together. Up here, we can see that there's one amino acid attached right here that has a long R group with a positive charge, and there's another amino acid way over here that has an R group with a negative charge. And those positive and negative charges are interacting, forming an ionic bond. And that is going to hold these portions together. Then down here, we have another example where maybe we have an amino acid right here and here that both have nonpolar R groups. So both of those R groups are hydrophobic, and they are going to fold together so they can create a small section that is going to stay away from water if this protein was in a inside the cell or in some other environment that had a lot of water. So these are called hydrophobic interactions, just folding together to stay away from water. Finally, we've talked about hydrophobic interactions, ionic bonding, hydrogen bonding. The last category is disulfide linkages. And this is a covalent bond between sulfurs. So there are amino acids that have sulfur as part of the R group, and sulfur is able to form a covalent bond with sulfur. And so here we have two sulfurs covalently bound together. Remember, that's a strong interaction, and so that could anchor this loop in place, for example. So the sum of all of these interactions will determine the tertiary structure of a single polypeptide. Now, let's think about proteins that have multiple polypeptides. So maybe there's a protein that has a chain like this, but then it also has a second chain. And the second chain also has some shape. I'm going to draw it way over here on the right, like so. These two proteins will come together, and they will have interactions between the two chains. I'm going to try to draw the same structure. I won't be able to get it exactly the same. There are going to be the same kinds of interactions between the two chains that will help hold them together in a particular orientation. So for polypeptides that for proteins that have more than one polypeptide, the structure that they take when held together is referred to as their quaternary structure. This says interactions between several polypeptides that make up a protein. Really, it just has to be greater than one, so two or more polypeptides that make up a protein. So that then reviews primary, secondary, tertiary, and quaternary structure. This diagram summarizes all four levels of structure. We've got the primary, just order of amino acids. Then we have certain sections that are forming beta pleated sheets or alpha helices. Then we have the complete folded shape of individual polypeptides, which includes their alpha helices and beta pleated sheets, but also all of the other folds. And then for proteins that have more than one polypeptide, we have the shape of those folded together as their quaternary structure. We've said that a protein's shape determines its function. That means if we can change a protein's shape, we can stop it from having its intended function. And we use this all the time, for example, in cooking and in protecting ourselves from disease. So the general term for taking apart the structure of a protein is denaturing it. We're taking it out of its natural shape. And we can do this in a few different ways. One way we can change a protein's shape is by changing the pH of the solution it's in. So we could either add or we could remove hydrogen ions. We could, in other words, lower the pH, or we could increase the pH. And changing either of those can change the ability for ionic interactions to occur, because the charges on R groups may change depending on whether the pH of the solution they're in is low or whether the pH is higher. Another way that we can denature or change the three-dimensional structure of a protein is by changing the temperature. Remember that heat breaks bonds. So if we add heat, we can start to break those bonds that hold together helices and pleated sheets. We can also break the bonds that are holding together tertiary and quaternary structure. And that then can cause the protein to not function. A third way we can change protein structure is by adding salts. And this will have a similar effect to changing pH. So we do this all the time when we are trying to preserve food, for example. When we make pickles, we're trying to stop bacteria from eating the pickles or reproducing on them. And so we do a few things. First, we heat the pickles, and so that's going to change the temperature and disrupt proteins and bacteria. Second, we add vinegar, and the vinegar is going to lower the pH. And third, we add salt, which is going to again change the possibility of ionic bonds forming. We also use heat to break down proteins when we want to be able to digest proteins. So for example, this fried egg, we are breaking down proteins as the egg cooks. And that is the end of our general discussion of proteins. We will of course discuss them more throughout the semester. Let's go over just some summary points that are important to remember. First, proteins have a whole host of different functions, and we enumerated several of those at the start of this lecture. Second, the monomer of proteins is the amino acid, and different amino acids have different properties because they have the variable group or the R group that may be polar, nonpolar, have a charge, or have a ring. And finally, the protein's order of amino acids is going to determine its primary structure, and that will determine its secondary structure, its tertiary structure, and its quaternary structure. And all of those structures together will determine an enzyme's function.